goal for this is to create the environment where in 24 hours, anybody with any background can build anything.
The 3 tracks were in closed-loop science, so using an agent to come up with media compositions to grow a new bacteria.
When we unlocked this hackathon, instead of just tweaking some liquid handling parameters, we actually opened up our full reagent library.
We're working with folks in hardware, we're working with folks with software, supporting different kinds of applications. What needs to come next? Where are there gaps? Working with these teams allows us to better understand that.
The second track was to streamline a single-cell isolation step that's used often for clonal engineering to make sure when you're growing cells that they all come from a single cell. And that was done via computer vision and microscopy-based sensing and liquid handling to do serial dilutions.
What surprised me the most this weekend was like how people were really using Elnora for things that I didn't know are possible. And I was like, wow, amazing. You're really pushing the limits.
So track C was to manipulate tea flasks, for which traditional lab automation is not yet well equipped. And so we brought in a general purpose 6-axis robot arm with a gripper to manipulate these tea flasks that are generally used manually.
This weekend, we built automation for bioscience. So basically, it's a combination of AI-driven task planning plus the robot actually execution of the task planned.
Optimize any code base, throwing bacteria, 10-minute doubling time. That's how real science got done.
AI transition, one plate, less than 24 hours, and I'll show you at the end.
That doesn't sell anything.
It is good, yeah.
We're doing an open source skill for Elnora, what we think about.
We're getting ready to do an AI for Science Summit. We want to open up a lot of the work that if you had to be in San Francisco to try this out, record the talks, share the work so more people can build.